Hello, everyone. I am Claudia Labrador. I'm the Global Product Manager for Reagents at Stilla Technologies, and I am very pleased to present you today Dr. Alexandra Lespagnol for today's webinar session. Dr. Lespagnol is a scientist in the Laboratory of Somatic Genetics at the Rennes University Hospital in France. The objective of this laboratory is to analyze tumor samples, either FFPE or liquid biopsy, to highlight mutations for diagnostic purposes. She'll be discussing with us today the main insights of her studies, screening pan-tumors mutations with using high multiplexing panels for EGFR and ESR1 for FFPE and liquid biopsy samples through digital PCR. So I give the floor to Alexandra, that she will share with us a very interesting insights.
Okay, thank you, Claudia. Hello, everybody. So before to discuss with you about the result of this evaluation on these two different panels, I just introduce and explain how the laboratory is organized and the objective of these laboratories, the activities. Okay, so the mission of the somatic genetics laboratories is to detect mutation, to do a molecular analysis on the tumor samples to adapt the therapy of the patient. We work on two different samples, FFPE samples and liquid biopsy samples. For these two different type of samples, the common is the low quantity of DNA and the poor quality of the samples. In France, we have to do molecular diagnosis on 15 different type of cancer, like lung cancer, breast cancer, or cholangiocarcinoma. We do this type of analysis with NGS part, or digital PCR, and or pyrosequencing. It's around 80 to 1000 different analyses per week. And we have to check mutation on two different type of genes, oncogenes and the tumor suppressor genes. On oncogenes, we have to analyze a very specific mutation. So the digital PCR is very adapted for this type of genes. And we keep the NGS for analysis of BRCA1 or BRCA2 genes. Okay, and we will do this diagnosis at just the beginning of the disease, and also after the treatment to try to understand the mechanism of resistance, the progression. We have, for the moment, three different applications for the digital PCR analysis. First is before the NGS analysis to try to detect EGFR, KRAS, or BRAF mutation for lung cancer or melanoma, for example, and also resistant mutation on ESR1 gene. And if this first analysis by digital PCR is positive, if we found a mutation, we don't necessarily to do more. We can stop here, and the doctor have the good answer and can adapt the therapy. And if it's non-mutated, we will follow by NGS. The second application of the digital PCR is to analyze the methylation of the promoter of BRCA1 and RAD51C in addition to the NGS analysis. For example, for ovarian cancer, we need to these two different analyses to identify the maximum of patients who are eligible for PARP inhibitor. And another application is to detect the presence of tumor DNA in liquid biopsy. It's important to know if in this liquid biopsy, in the cell-free DNA, we have tumor DNA. And for example, for colorectal cancer, the presence of WIF1 or NPY methylation in the liquid biopsy sign the presence of tumor of DNA. In the case of this analysis is positive, we can follow by digital PCR or NGS, and if it's negative, we have to stop it. It's not necessary to lose time. After the activity of the lab, one thing important is how we organize the workflow to try to keep time, to reduce turnaround time, to save the the samples and also to facilitate the workflow. First is the automation. So in the lab, we have a Hamilton platform, STAR, and we develop different pipeline, different program to do all the steps of the preparation of sample, like extraction of DNA and cell-free DNA, the preparation of plate for quantification by qPCR, and also the deletion of the DNA at the good concentration. Okay, and we will do all these steps in only one day. And because the Ruby chip is compatible with multi-channel pipette, we decide also to implement in this workflow also the preparation of plate, the mixed PCR, and the loading of Ruby chip by the same platform. The objective is to standardize the workflow and also saving time. It's very important in somatic laboratory. The second point is the NIO+ system. I have the opportunity to test this equipment during the evaluation, and in the lab for the moment we have two geodes and one Prism 6. The NIO+ system is an all-in-one platform. So on the same equipment, we will do the partition and the amplification PCR and also the reading. You have two different thermocyclers on this machine, independent thermocycler, and you have also an automated plate queue for to manage 8 different plates. So it's easy and it's better for the technician. If you share the digital PCR platform between different teams, it can be important to have this type of equipment. The technician who did the plate just focus on the plate and come to the machine, put the plate on the machine, and he have the real time of the ending of the analysis. So it can be better for the organization, and you can increase the number of plates per day. Okay, another important plate, it's a platform with 7 different colors to increase the multiplex. The next point is the new concept. Okay, so if you use a usual approach for the digital PCR, you have one color per target, and depending on the number of color you have in your machine, so 7 for example for the NIO+, you can mix together 7 different targets. With this new approach, the color combination approach, you have 2 different colors per target. So you increase the number of combination, so you increase the number of targets in only one panel. Okay, it's 21 different targets for panel running on the NIO+, and 15 on the Prism 6. The other important point is during the analysis on Crystal Miner or NIO+ analyzer, you do different settings to only analyze the droplet with the 2 different colors. So like we will see in the next, you reduce a lot the background level of the analysis. It's very, very important point for FFPE samples. And the new concept more is you have 2 different colors on one target, but the fluorescent molecule are moved from the target to universal reporters. Okay, if you need more information, I think it's better to discuss with Stilla team. But for a user, I think very different advantage to this new concept is to reduce the time to do the design and to finalize a panel, because you only order a primer without fluorescent. You also reduce the cost of the design. And one important thing, these universal reporters are compatible for all the panel you will have in your lab. So you can imagine 2, 3, or 4 different panels in your lab, but you have only two reference, one for the mix, one for the universal reporters, and only the panel. So you simplify also the work on the bench. And the last point, because you can increase the number of targets, the number of samples in the NIO+, you can increase also the number of targets with the new concept. And so you have a Excel file very, very big. You do the analysis on NIO+ analyzer or Crystal Miner, and you export your Excel file. The Excel file is depending of the number of samples in terms of lines, and also is depending of the number of columns, depending of the number of targets. So I'm not really fan of this tape to transform numerical data in biological data, because we use the digital PCR to do diagnosis. Okay, it can be very difficult, and the error is easy. So we develop with the bioinformatics teams of the hospital dashboard and interface to help to the interpretation and the validation of digital PCR result data. Okay, so we combine in the same interface the results of the NIO+ analyzer, for example. Also the quality criteria of digital PCR, for example, the number of partition or the number of positive droplet. We also inject the decision rules, so which value I have to use to calculate, for example, the concentration, or to calculate the allele frequency of the mutation, and also to transform a non-mutated on a mutated result. So all the rules are included in the interface, and also we inject from the LIMS different information, clinical information, to have the context of analysis. For example, the number of percentage, the histology type, or the cell content. So with this type of interface, we can analyze the run in three different steps. First, the run quality of the puce. Okay, first check this is an overview of the chip. So you will see all the samples and also the QC of the chamber and the QC of the samples in the chamber. It reflects the number of partitions and also the number of positive droplets. The second part is to validate the positive control and the negative control, the blank. There is a contamination or not for each different target in the blank, and also I found all the expected mutation in my positive control. And if the run is okay, if the positive control and negative control is okay, I can start to analyze my sample. So in this part is two different, it's an example with two different samples. One sample is mutated, so the interface with the rules transform this information, 260 positive droplet in this type of mutation. Okay, this part is an automated validation, but the last step is under the responsibility of the biologist. Okay. Oh, and this type of samples is a non-contributive samples because you don't have enough DNA in the samples. So at the end of the interpretation of the interface, you have an Excel, the creation of an XML file to send the result in the LIMS to start to create the report. So we facilitate this step. So now result about for the EGFR result. So we evaluate this kit, commercial kit, 6 color for EGFR with around 100 different samples. This panel is designed to detect 7 different mutations, sensitive mutation and resistant mutation in 4 different exons. Is for lung cancer screening for the EGFR tyrosine kinase inhibitor. We evaluate with a sample with different tumor contents, different DNA concentration, and also a different genotype. This genotype was obtained with the NGS in the lab. Just for remember, with the usual approach, we have to select the positive population with a polygon. So it's easy to do that on the positive control and also on cell-free DNA, but it can be sometimes a challenge for FFPE samples because depending on the quality and the quantity of DNA you add on your chamber, you can sometimes not a real difference between two different populations. So the results are very good. We have 100% of sensitivity on FFPE samples and a specificity of more than 98%. We detect one more mutation with the digital PCR compared to the NGS. We also compare the allele frequency between NGS and digital PCR. We have a very good concordance with 0.93. And we also check the repeatability on the positive control. We have a little variation different between the value, but it's very acceptable for the diagnosis. The other point, we calculate the limit of blank for this kit on FFPE sample. The data are present in the copy per microliters. It will be better to do this in percent. But the background, we observe a little background. It's under 1% for these samples. The next evaluation was ESR1 assay. So it's an assay with color combination approach. So is designed to detect 10 different mutations. Okay, and the specific mutation is not a drop-off. You can identify each mutation specifically. They also add a AKT1 mutation, and the gene to calculate the allele frequency is an housekeeping gene, the TSN. This assay, designed by Stilla, covers more than 80% of the ESR1 mutation described in the PADA1 study. We evaluate this assay on FFPE samples and also on cell-free DNA, like for EGFR. We test different tumor content, different DNA concentration, and different genotypes. Notice that on the cell-free DNA, we don't know the genotype before to do the digital PCR. And we also do a new run with the same sample to evaluate the repeatability on the real sample. So for the analysis of this type of this new approach, you just have to fix the threshold for each panel to simplify really the analysis compared to the usual approach. And you add different parameters in the NIO analyzer to just analyze the double positive droplet. Okay, and so you do this analysis in the one dimension representation, but you also can check your positive population in 2D plots. So the results are very, very, very good. We have 100% of sensitivity and specificity between NGS and digital PCR for FFPE samples. The allele frequency is very concordant between the two technology, and the repeatability is quasi perfect because there is a very, very low difference between the two different experiments. And the LOB of this ESR1 assay is calculated like a EGFR. We exclude mutated samples, non-contributive samples, and also we exclude over and under concentration just to keep the samples. And on 74 FFPE samples, the background can be is under 1 copy per microliters and often equal to 0. So we have a very good effect of this new technology on the background with FFPE samples. We also analyze the cell-free DNA. So we identify 5 different samples with EGFR1 or AKT mutation. So we do the experiment in 3 plicate. And here in this middle of the schema of the middle, we can observe the value of the 3 experiments, and sometimes we don't have a difference between the 3 different experiments. So we also have 3 non-contributive samples because we don't have enough DNA. So it's not magic. If you don't have DNA, it's not possible to do a diagnosis. It's important also to identify these samples during the process of validation. So the first result was very good, but now we try to challenge the Ruby chip and ESR1 assay. It's a really preliminary result. We start this challenge last week. And first, it tried to define and to challenge on the DNA amount, try to be in the same condition than the liquid biopsy. The cell-free DNA is not very concentrated. So we just use mutated samples we diluted in water, and we do we check the mutation, this mutation, and we observe the presence of the mutation on all the samples between 3 nanograms and 0.3 nanograms. So the mutation is detected. Just for remember, 3 nanogram is around 1,000 copies, and 0.3 nanogram it's around 100 copies. And the second challenge is try to dilute the mutated samples in non-mutated DNA to try to know if at low allele frequency the digital PCR detect. All the experiments are done in the Ruby chip. So we can detect the mutation at 3%. It's not challenge. 1.5%, also 0.7%. And at 0.3%, we detect the mutation. At 0.9, we also detect the mutation in the samples, and only in one chamber. We don't do a replicate for this analysis. And the last experiment was done on cell-free DNA. First, this cell-free DNA was analyzed by NGS, and after we did digital PCR. So the first samples is mutated samples detected by NGS at 8%, and we found the same mutation at 10%, correspond at 33 positive droplet. The second sample was negative by NGS, but by digital PCR, we detect a mutation at 0.5%. It's 10 positive droplet. And just to be sure, we also analyze a negative samples, and for this mutation, the background is at 0. We don't observe a background at this mutation. So these first results are very encouraging for the next step. So in summary, the new workflow for digital PCR is it's a combination of different aspects, different steps: the automatization for simplification and reduce error and facilitate the bench job. The NIO Plus is, I think, it's a facility when you share the equipment between different teams. The new concept is a very smart technology to simplify, reduce the cost, and also in somatic, it's very important, reduce the quantity of DNA used to do the analysis. And also, if you can increase the number of samples, if you can increase the number of targets, you need to facilitate the analysis by a dashboard, for example. So to making a digital PCR more accessible for everybody. Thank you very much.
Thank you very much, Alexandra, for your time and for this of your study. We open the floor for some questions. Um, that we will still have many questions that may not get addressed throughout this live session, but please, and I encourage you to publish them through the chat. We will get to them via email if they cannot get addressed during this live session. So I'll go ahead and share some of the questions that we have on the chat. So starting for the ESR1 assay, the question is like if you use drop-off assays, and if so, is that all 10 mutations or just certain ones?
No, it's not a drop-off technology. It's the sorry. I heard me. It's a specific design to detect each mutation separately. It's not a drop-off. Not sure to answer good to the question.
Thank you very much. So we have a next question. How much input DNA amount for at EGFR and the ESR1 assay?
Alors, for us, because we dilute the DNA we use 3 nanogram of DNA per chamber. But we challenge to try to reduce more the quantity of DNA because sometimes we don't have enough samples.
Perfect. The next question is, finding a suitable positive control can be challenging, especially when you're working with clinical samples. Can you work around this by using other type of sample sources, such as cell lines known to carry mutations of interest?
I'm not sure to well understand it. If I well understand, the positive control is fourni supplier by with the assay, but I'm not sure to understand well.
Okay,
that's correct.
A question. Yeah, thank you very much. So for how much DNA was used in the comparisons with the NGS results? For example, for the assay showing an 80% variation of fraction in NGS and 10% in dPCR, showing 33 positive partitions.
I think there is not a lot of difference between NGS and digital PCR because we use amplification technology for the NGS, and we use, I think it's around 5 nanogram of DNA. So it's likely the same, but the NGS is not a quantitative validation, it's just a qualitative for qualitative technology. So I know we have access to the quantitative value, but we don't do the real checking to be sure the allele frequency we observe in NGS is the correct one. I know we have a little variation between the two technology.
So in that same topic, this will be our last question of today, since we're running out of time. But we will get back to you for the other questions in the chat via email. So regarding also NGS and digital PCR, for the comparison between those techniques, in liquid biopsy, you analyze only 3 nanograms in NGS and 3 nanograms in digital PCR for one chamber?
Yes.
Okay. So then that will be our last question of today. Thank you very much, your audience, and thank you very much again, Dr. Español, for sharing the insights about denial capabilities. If you have questions or you're interested in learning more about all-in-one denial plus features, its continuous loading, the 7 detection channels, or its automation friendliness, as well as the color combination approach, do not hesitate to contact us or visit us at stillatechnologies.com. Thank you, everyone, and I wish you a lovely afternoon.
Thank you.